Today we are going to discuss about Salmonella, which is a gram-negative pathogen. We will particularly focus on the molecular aspects of pathogenesis of this bacteria. Now, the principal clinical syndromes that are associated with this bacteria, say in human, are of two types. First of all, the enteric or typhoid fever, and secondly, the more common gastroenteritis. Now, the enteric fever or the typhoid fever, it is a systemic disease. It causes systemic infection and it results from the typical human specific pathogens Salmonella typhi and Salmonella paratyphi. The clinical manifestations include fever, abdominal pain, transient diarrhea or constipation even, and occasionally a maculopapular rash. The typical hallmark of this typhoid fever is infiltration of the mononuclear cells, for example, macrophages. It is associated with the hypertrophy of the reticuloendothelial system, including the intestinal Peyer's patches, mesenteric lymph nodes, spleen, and bone marrow. Now, without treatment, the mortality can go up to 10 to 15% of infected individuals. Now, most of the Salmonella strains are non-typhoidal. The most well studied are Salmonella enteriditis and Salmonella typhimurium. They usually cause self-limited enteritis in humans, which are featured by intestinal inflammatory response as well as rapid secretory response, which is called flush diarrhea. Now, most of the Salmonella, whether it is typhoidal or non-typhoidal type, they begin their infection through the oral route, like when you ingest the pathogen via contaminated food or water. Now, the first barrier that the bacteria encounters is the gastric acidity. Now, Salmonella has a typical adaptive acid-tolerant response via which it can resist the acid stress that it encounters in the gastric juice. Now, after entering the stomach, if it survives, it will pass to the small intestine. And when it enters the small intestine, it can traverse the intestinal mucus layer using the flagellar motion. When it reaches the intestinal epithelial cells, it will interact with the intestinal epithelial cells and will adhere onto it via its fimbriae-like structures. Here we can see in this figure that this is an atomic force microscopic image of the Salmonella typhimurium. Say here you can see the flagellar structure. In this magnification, the fimbriae is not visible, but in a higher magnification, we can see that these smaller structures, they represent the fimbriae. These are the structures by which Salmonella adheres to the intestinal epithelial cells. Now, when Salmonella adheres to the intestinal epithelial cells, it will get internalized into the epithelial cells to cause the infection. So, this is done by a process, a typical process called bacterial mediated endocytosis. This is a scanning electron micrograph of this process. You can see that, say, this is the bacteria which has adhered to the cell surface. These are the structures which we call membrane ruffling. The membrane has engulfed the bacteria like this. Say in this case, you can see the bacteria is surrounded by the membrane, and say this bacteria is already almost fully internalized or engulfed by this process. So this is a typical process which is different from receptor mediated endocytosis.
Sir, what causes the dynamism in the membrane structure of the intestinal epithelial cells?
This is actually involves a profound cytoskeleton rearrangement of the host cell. This requires a coordinated synthesis of multiple bacterial proteins and specific host cell signal transduction pathways that regulate the cytoskeletal reorganization. Now, this is sort of an overview of the enteritis, the gastroenteritis done by the non-typhoidal Salmonella. You can see that the bacteria is being internalized by the membrane ruffling process, and when it enters the cell, it causes release of cytokines such as interleukin 8. This interleukin 8 then signals the recruitment of the neutrophil cells to the other side. These neutrophil cells then traverses through the intestinal epithelial cells to the other side, that is the intestinal lumen. Now, this results in the intestinal inflammatory response that is observed during non-typhoidal infection. This also causes rapid release of fluid such as water and a lot of electrolytes shown here like chloride to the intestinal lumen side, resulting in the flush diarrhea. This is the case of the enteric fever, that is the typhoid fever. Here the situation is quite different. After the internalization via membrane ruffling, the bacteria crosses the intestinal epithelial cell barrier and passes to the other side. Here you can see that they are being internalized by the macrophages that populate this phase via a different process. This is receptor mediated endocytosis, and in this case toll-like receptor or TLR receptors are involved. Now, normally a regular bacteria will be destroyed after engulfment within the macrophages by the different macrobicidal actions of the macrophage. But in this case, as you can see, that the engulfed bacteria survives. Not only survives, it can even replicate within the macrophage. And then the macrophages themselves, they act as carrier of this bacteria, and they take it to the reticuloendothelial system via the lymphatics and the blood, and thereby they can cause the systemic infection. Now, if we look at the different virulence mechanism of Salmonella, we can see that most of its virulence factors are encoded by different pathogenicity islands, which are obviously acquired by horizontal gene transfer, like regular pathogenicity islands. Now, you can see here there are two major pathogenicity islands in Salmonella: Salmonella pathogenicity island 1 or SP1 and Salmonella pathogenicity island 2 or SPI2. This is the organization of SPI1, and this is the genetic organization of SPI2. Here you can see that the color codes, they represent genes of related functions. As you can see here, the genes of related functions, both in this SPI1 and SPI2, they are sort of clustered together. Now, SPI1, they encode genes necessary for invasion of the intestinal epithelial cells, as well as induction of the observed secretory and inflammatory response in the intestine. On the other hand, SPI2, they encode genes essential for intracellular survival and replication, as well as establishment of the systemic infection beyond the intestinal epithelium. Now, it is interesting to know that both SPI1 and SPI2, they encode genes for type 3 secretion system, that is the device that is used by the bacteria to inject its own virulence factors inside the host cell cytoplasm and thereby modulate the cell biology of the host cell in its own favor. So, this is a schematic representation of the type 3 secretion systems encoded by Salmonella and many other gram-negative bacteria. See, this part is the bacterial part and this part is the host cell part. The two membranes of the bacteria are shown, the outer membrane and the inner membrane, and this is the cell membrane of the host. There are two ring-like structures at the base of this machinery, and this is connected to the host cell membrane via a needle-like structures. Via this machinery, Salmonella virulence factors are injected inside the host cell cytoplasm, where they will regulate the host cell functions. Now, there are many different effector proteins or virulence factors that are delivered via these microinjection systems inside the host cell. If we consider the factors involved in the inflammatory action as well as the secretory action that we see in the case of enteritis, we will see that there are several effector proteins. For example, SOP-E and SPTP, which target the Rho family member G proteins of the host cell. Examples are CDC42, RAC, Rho, etc. Now, these Rho family G proteins, they regulate gene transcription by stimulating signal transduction via the MAP kinase family members of the host cell, that is the mitogen activated protein kinases. And all these downstream effectors, they regulate the rearrangement and reorganization of the host cell cytoskeleton, and thereby regulate the internalization of the bacteria within the intestinal epithelial cells. Other such effectors injected by the type 3 secretion system are CYP-A and CYP-C. They interact directly with the host cell actin cytoskeleton. They act in concert with the downstream cellular effectors of the Rho family GTPases, and thereby regulate the initiation and localization of the rearranged actin filaments to form the membrane ruffling structures and result in the bacterial mediated endocytosis. There is another factor called SOP-B that actually promotes the intestinal inflammation and fluid secretion, that is results in the flash diarrhea, by subverting the inositol phosphate signaling pathways. Now, if we look into the other mode of infection by the typhoidal type of Salmonella that causes infection of the macrophages and systemic infection, we will see diverse type of virulence factors. First of all, there is a two-component response regulator of 4P, 4Q. Now, this 4Q is the transmembrane sensory kinase that senses the entry of the bacteria inside the phagosome, and thereby it phosphorylates the downstream effector in the cytosol, that is the 4P component, and this 4P after phosphorylation gets activated and act as transcription regulator. Now, these 4P activated genes, they are expressed within the macrophage phagosome, and these expressed genes are required for the survival of the bacteria within the macrophage. For example, 4P leads to covalent modification of the lipid A component of the lipopolysaccharide of the bacteria, which results in the resistance towards the microbicidal action of the antimicrobial peptides produced within the macrophages. Then, of course, there are virulence proteins or effector molecules synthesized within the bacteria and translocated into the cell by the SPI2 type 3 secretion system. So, this system also gets activated only when the bacteria is within the phagosome, and then it causes its delivery function in the macrophage cytosol. Now, these effector proteins, they usually interfere with the maturation of the Salmonella containing phagosome. For example, it inhibits the fusion of the lysosome with the phagosome and thereby allow the Salmonella to physically avoid the microbicidal factors within the macrophages. For example, SPI2 type 3 secretion system prevents the co-localization of Salmonella containing vacuoles with the NADPH dependent oxidase within the macrophages, which results in the formation of a rapid burst of reactive oxygen species. This is sort of an overview of how the bacteria survives the macrophage environment. You see, after being internalized by the toll-like receptors, they ultimately can survive the acidic pH of the phagosome, and finally they can even replicate within the phagosome, and during that process they inject out by the type 3 secretory system the different SPI2 encoded effector molecules by which they can modulate the macrophage cell biology.
Sir, how does Salmonella inside the phagosome survive the reactive nitrogen and oxygen species?
Well, Salmonella itself, they express several enzymes that can directly interact and inactivate these highly reactive oxygen and nitrogen species. For example, one of the strongest reactive nitrogen species that is active against engulfed bacteria within the macrophage is nitric oxide, or NO. Nitric oxide attacks the bacteria, in this case Salmonella, in two ways. First of all, being highly reactive, it will chemically react with the different macromolecules within the bacteria and thereby kill the bacteria. And second of all, the nitric oxide can inhibit the expression of SPI2 encoded genes and thereby it can promote the killing of the bacteria. Now, Salmonella counteracts this nitric oxide attack by two ways. First of all, it uses this nitrite transporter called NEED-C to import nitrites within the bacteria, and these nitrites, they can quench nitric oxide very well. Second of all, they use SPI2 expression to produce an effector molecule SPIC that they translocate via the secretory system into the macrophage cytosol, where this SPIC can inhibit the expression of iNOS, which is the nitric oxide synthase, and thereby prevent the synthesis of nitric oxide. In these two ways, they can neutralize the action of nitric oxide. Now, for reactive oxygen species, Salmonella can express various superoxide dismutases, or SODs. Normal typhoid causing Salmonella produces only one superoxide dismutase, but hypervirulent strains, they express two superoxide dismutases by which they can neutralize the reactive oxygen species within the macrophage and survive and ultimately cause the expression. Now, one more stress that the Salmonella encounters within the macrophage is nutrient limitation. Now, to counteract that, it uses another strategy. It induces the expression of multiple biosynthetic genes. For example, those necessary for the de novo synthesis of the essential metabolites like aromatic amino acids and purines, and thereby it produces its own nutrients necessary for its survival. So this is the key. If the bacteria can survive within the macrophage, it will be able to travel via the macrophage using the lymphatic and blood system to infect the rest of the body. Now, that is sort of end of today's discussion. If we look back, we will see that Salmonella, the typical gram-negative pathogen, it causes two types of human infection. One is the common gastroenteritis, which is accompanied by intestinal inflammatory response as well as secretory response, that is flush diarrhea. This disease is not so fatal. It is usually self-limited, and the body takes care of it within 4 to 5 days. And then we have the other more virulent form, that is the typhoid fever or enteric fever, which is caused by the specific human pathogen Salmonella typhi and Salmonella paratyphi. This causes systemic infection, and if untreated, then it can cause fatality. Now, to achieve this virulence, all the different types of Salmonella, they use several virulence factors. First of all, they have their own processes like flagella and fimbri to adhere to the intestinal cells, and then they can internalize the intestinal cells and infect it. And the non-typhoidal type will do the enteric response, and the typhoidal type will further cross the intestinal barrier to infect the macrophage and result in the systemic infection. They use many different virulence factors that are encoded by the two pathogenicity islands, and both of the pathogenicity islands use type 3 secretion systems, by which the bacteria can deliver various effector molecules that ultimately will enter the macrophage cytosol and even the other intestinal epithelial cell cytosol, and thereby modulate the cell biology of the host cell in